One of the most stunning scientific moments of the last decade occurred in 2012 at MIT. The big news in the scientific community is that a team of scientists was able to implant a false memory in the minds of mice. Imaginations are running wild with the possible future implications.
I know Kung Fu.
The idea of memory manipulation is truly fascinating, but the technology that was integral to this breakthrough promises to influence every corner of neuroscience. Optogenetics is a method used to target very specific neurons in the brain, which is helping scientists gain a superior understanding of brain function. Older techniques of brain stimulation comprised of electrical wires hooked directly up to the brain that could be turned on and off to stimulate neurons. This crude method would activate all neurons in an area and gave only the blurriest picture of brain functions. To understand optogenetics, we first need a little knowledge of the architecture of the brain. We tend to think of the brain as a collection of evenly spaced identical neurons swimming around in the skull. This couldn't be further from the truth. The brain is made up of many, possibly thousands of different types of neurons, each with their own unique function and genetic code. If we could activate only one type of neuron within a region of the brain, we can track brain activity much more precisely. Think of electrodes as a 5-pound hammer, while optogenetics is more like a pair of tweezers. For example, electrode studies identified the regions of the brain responsible for memory, but through the use of optogenetics, a group at MIT was able to record the formation of a specific memory in a mouse and replicate it later in the brain of a completely different mouse. So how does it work? Certain algae have a built-in light sensor that allows them to seek out light for photosynthesis. A protein called channel rhodopsin reacts to blue light stimulation by releasing electric potential. The code for this protein can be inserted into the genes of a particular type of neuron, so that when the brain is illuminated by a specific frequency of light, the treated neurons and only the treated neurons will fire. This allows research teams to turn individual types of neurons on and off with millisecond precision. Other similar proteins react to different wavelengths, and some can be used to inhibit rather than stimulate the firing of neurons. So who cares? What is this going to change in the field of neuroscience? The answer is everything. This is like introducing a scalpel to surgeons who've only ever used chainsaws. Optogenetics is not going to make all of our discoveries for us, but it is a major step in the quest to understand the human brain. In addition to fueling new discovery, it seems likely that we will be able to treat some neurological problems by directly interfering with the brain. We already use electrical stimulation to combat epilepsy, so the possibilities for this much more sophisticated technology are staggering. Maybe a long time, if ever, before we were able to implant specific memories. So for now at least, if you want to learn Kung Fu, go to a dojo.